phytoplankton also play an important role in the biological pump. The biological pump is the mechanisms through which carbon is taken up by the ocean. And how this happens is that phytoplankton take up carbon dioxide as they photosynthesize. And eventually they die, and the cells containing the organic matter sink down to the ocean interior, where they get stored for a very, very long time. So to understand the effects of phytoplankton, both on the food chain and on the carbon pump, it is actually very important to understand what affects their growth. And there are three main factors. Temperature is one of them. As most microbial metabolism, phytoplankton growth or primary production is expected to decrease with temperature. The second factor is light. Because they photosynthesize, they need light to do so. And in the ocean, we have what is called a mixed layer. The mixed layer is a surface layer of the ocean that gets mixed up by the wind. And as the surface layer gets mixed, phytoplankton moved up and down. And this affects the amount of light that we receive. So if we have a deep mixed layer, phytoplankton will receive less light. And the third factor that I'm going to talk about here are the nutrients. So phytoplankton need nutrients such as nitrate and phosphate to grow. And they also need micronutrients such as iron, which is what we're going to focus in this talk. In the Southern Ocean, there is a lot of nitrate and nitrite and phosphates. But as you can see from this picture, the chlorophyll concentrations in the Southern Ocean are quite low. And the reason for this conditions that we call high nutrients, low chlorophyll, is that there are actually very low concentrations of iron in the Southern Ocean. So in this figure here, you can see that the blue, which is represents low iron concentration, affects most of the Southern Ocean. And because of this iron limitation, phytoplankton in the Southern Ocean can't grow as much as we would expect given the high nitrate concentrations. So what we did in the study was to try and see what happens when we add iron. How much more do phytoplankton grow? And what we did, we set off on the SA Gullis 2 in January, and we went to 2 stations, one at 46 South and one at 65 South. And we collected water samples and we separated them into 6 lots. So half of the lots had no iron added to it, and we added iron to half of the bottles. And then we incubated them at 3 different light levels. So we had L1, L2, and L3, which were the lowest was L1 and the highest was L3. And then we measured FV over FM, which is a measure of photosynthetic efficiency, which means how well do the phytoplankton use the light that's available to them. We measured chlorophyll concentration, as this gives us an indication of how well and how fast they're growing. We also measured a whole lot of other parameters, such as nutrients, to see how well they're using the nutrients. We measured pigment and genetic samples to have a better idea of what phytoplankton are in there, which species are in there. And particulate organic carbon is also expected to give us a measure of the growth. So onto our results. If we look at the FV over FM, which is how well the phytoplankton are able to use the light, we can see here that they all started at a very low FV over FM. And this is expected in the Southern Ocean because those phytoplankton are limited and they can't grow as well as they can't grow very well. And here we see that at our station at 46 South, the increased light, so the highest light level, created the largest growths, the largest increases in FV over FM. And this is even clearer when we look at our total chlorophyll concentrations. Here we see that L3 in both with added and no iron were actually produced the highest growth, whereas L2 also increased the growth, but there was no differences between added iron in L2 and no iron in L2. But how is this different to our results from the samples at 65 South? Sorry, I keep going wrong way. At 65 South, we saw that our added iron is what increased our photosynthetic efficiency the most. So you'll see the dashed lines here, here, and here. They are all represent our added iron, and all of these are higher than the no iron samples. And again, this is even clearer in the chlorophyll concentrations, where we see both samples with added iron were much higher than the samples with no iron. And this applied even if the light, say at L2, was lower than the light at L3. And the main question here is, why are those results so different? And the reason for this is the community structure. So what this means is what phytoplankton were actually living at those two different stations. As I first mentioned, phytoplankton exist in a variety of shapes and sizes. And what we have here are the different sizes and the amount of chlorophyll in each size class. And what we see is that our station at 46 South had a high proportion of smaller phytoplankton, but our station at 65 South had a higher proportion of large phytoplankton. Another factor to note is that the chlorophyll concentrations here are on two different scales. Here it goes up to 0.16 micrograms per liter, whereas here it goes up to 2 micrograms per liter. So what this tells us is that at 65 South, the phytoplankton have had more time to grow, and they probably had used up a lot more iron and were therefore more iron limited than the phytoplankton at 46 South, where there were fewer phytoplankton